Hello everyone, my name is Rishabh and I will be talking about the Trypanosoma species. So in our content we will be looking at a bit of introduction, we will go over the pathogenesis, the life cycle, the lab diagnosis, the treatment, prevention, control, and then finally we'll end with our references. Looking at the introduction, we will first go over the domain, which is the Eukaryota, then comes the kingdom Protista, the phylum Euglenozoa, the class of Kinetoplastia, following in the order of Trypanosomatidae, family Trypanosomatidae, and the final genus, which is Trypanosomas. The various species are listed uh, below. This presentation itself will revolve around the Trypanosoma brucae and Trypanosoma cruzi due to the high lethality rate in the known species. Now moving on to the etiology. The African trypanosomiasis, which is the sleeping sickness, is caused by the Trypanosoma brucae. It is also caused by its two subspecies, which is TB gambians and TB rhodescens. The American trypanosomiasis, also known as the Chagas disease, is caused by Trypanosoma cruzi. Also, and we have another which is animal trypanosomiasis, which is caused by the following, including of Trypanosoma vivax. Moving on to some epidemiology. So for African trypanosomiasis, it is predominantly found in sub-Saharan Africa, spread by tsetse flies of the Glossina species, and some risk factors include rural areas, water bodies, occupations involving outdoor activities in endemic regions. Moving on to American trypanosomiasis. It's predominantly found in Latin America, but spreading is possible through migration. Transmission, it can be transmitted, mainly transmitted from triatomine bugs, which are also known as kissing bugs, also from congenital transmission, blood transfusion, organ transplants, and consumption of contaminated food and beverages. Some risk factors include poor housing conditions, rural areas, and high prevalence of areas where there is a presence of triatomine bugs. Next comes animal trypanosomiasis. This is particularly in Africa, Asia, and Latin America. So this can be spread by T. evansi by biting flies, and T. vivax and T. congolens by tsetse flies. So some risk factors include the presence of some vectors, livestock, and also in certain living conditions. Now moving on to the pathogenesis and life cycle of Trypanosoma brucae. So what happens is that a tsetse fly, it comes and injects a human in with the following virus, which is an Trypanosoma brucae. So what happens during the human stages is that the virus itself, it enters into the bloodstream and then it starts to undergo proliferation, not proliferation, but it undergoes multiplication by binary fusion in various body fluids like the blood, lymph, or the spinal fluid. So after that, its final stage is that it reaches a diagnostic stage where symptoms can be detected if proper testing is administered. Now moving on to when another fly, a tsetse fly, it comes and takes the bone, a blood meal from that infected human. Now during that stage of development, what happens is that in the bloodstream of the fly, it transforms into procyclic trypomastogotes in tsetse flies in its midgut. Now it undergoes binary fusion there where it multiplies, and then this stage continues where it multiplies also in its salivary glands, and it transforms into metacyclic trypomastogotes where it then reaches into the final stage where it becomes infectious. And then this cycle continues on where the fly comes and injects, or it takes a blood meal from a human, and then the cycle repeats itself. Now similarly in the Trypanosoma cruzi, what happens here is that the bug takes a sample of blood, not a sample, but it feeds off of the human and injecting the parasite. the parasite undergoes the same similar processes to that of Trypanosoma brucei. So it injects into the bloodstream and then it undergoes binary fusion in cells, infecting some tissues, and then it reaches a diagnostic stage. Now from there again a bug comes, takes a blood meal, and then the parasite moves into the bug itself. Now from there it proliferates in the midgut where it undergoes binary fusion and then it reaches its final stage which becomes the infective stage. Moving on to some laboratory diagnosis. Now the tests carried out and diagnosis for Trypanosoma brucei. So the first test being blood smear examination. Microscopic examination of Gamsa stained smears, and its diagnosis is that it detects the level of trypanosomes and the parasitemia level, which indicates the severity of the infection. Next is cerebrospinal fluid examination. Lumbar puncture followed by microscopic examination of CSF. Now this detects for the presence of trypanosomes in the CSF and also for elevated white blood cells. This indicates the presence of inflammatory cells in CSF, also indicating meningoencephalitis. Moving on to the next test, which is serological test. This includes ELISA, immunofluorescence assays, and RDTS, any test that targets specific antigens. Now the detection of antigens in response to the infection and the presence of IgM and IgG antibodies to determine the stage of the infection. The fourth test that we have is polymerase chain reaction. This is the molecular amplification of T. brucei DNA in blood or tissue samples, and this detects for parasite DNA and also helps in identification of the specific species type. Next we have xenodiagnosis, which is feeding uninfected tsetse flies with the patient's blood and followed by microscopic examination of flies. This for the presence of trypanosomes. Now the detection of trypanosomes is in the midgut of the fly, so if they are detected we can diagnose that the patient has infection of trypanosomes. Next, moving on to the tests carried out and diagnosis for Trypanosoma cruzi. First is a microscopic examination. So similar to the previous one, we will microscopically examine Gaim's stained smears. Now this will also detect for trypanosomes and the parasitemia level, which will indicate the severity of the infection. We will look at the blood culture, which will be inoculation of blood samples into specialized culture medias, and for parasitic isolation, where the growth of Trypanosome cruzi confirms the infection, allowing for further characterization. Similarly, with serological tests, we will conduct ELISA or IFA or RDTS, any of these tests which will help to detect antibodies that are produced in response to the infection and also for the presence of IgM and IgG antibodies. Polymerase chain reaction. These are molecular amplifications of T. cruzi DNA in the blood or tissue. Now this also detects for parasite DNA and also helps in identifying the species. Xenodiagnosis, we will be feeding uninfected triatomine bugs with blood samples from the patient, and we will follow up with microscopic examination of its feces, and the presence of trypanosomes in its feces will confirm that the patient has an infection. Treatment for human African trypanosomiasis. In Gambian's head, we can use pentamidine, which can be administered intramuscularly. This is administered during the early stage of the infection. Next comes eflonithine, which can be given intravenously. It's much safer than melarsoprol, but this is only effective in Gambian's head. Generally, it's co-administered with nifurtimox in a therapy known as NECT, but it can also be used as a monotherapy. Nifedimox, it can be given orally in during the second stage, and it's a component of NECT, which is also a shorter treatment for with 4 times less fewer of eflornithine infusions, and it's also safer and more effective than eflornithine alone. Next comes fexinidazole, which can be given orally in the first stage, and if the second stage is non-severe, this ensures efficacy to be taken after a solid meal and under supervision of a trained medical staff. Now, during the second stage, which not the second stage, sorry, the second type of African trypanosomiasis, which is rhodesians. Now we can give suramin, which is administered intravenously, and this is during the first stage, but this has some adverse effects like nephrotoxicity and some allergic reactions. Next is melarsoprol, which is administered intravenously, and this is given during the second stage, but it also has many adverse effects because it is an arsenic derivative, with its most drastic being the reactive encephalopathy, which has a 3% to 10% fatality rate. Moving on to the treatment of Americano trypanosomiasis, it can be treated with benzidazole or nifurtimox, both of which are effective in killing the parasite and curing the disease only if it is given during the early acute phase. Otherwise, they lose their efficacy the longer a person is infected. And also, these drugs have an adverse effect on elderly individuals. And for those individuals that show no symptom, they should be offered immediate treatment because antiseptic medicines, they also prevent or curb the disease progression. Moving on to the prevention and control for trypanosomiasis. Some ways that we can prevent is first by educating the community. We can develop information, education, and communication materials and bring up scenarios where we can allow for critical thinking, where people themselves they give ideas on how we can prevent the spread of infections. Some are by the spraying of surrounding areas with pesticides, which can kill off the bugs or flies that's surrounding the household. Regular cleaning of the home can prevent vector manifestation. Personal hygiene measures by the way we prepare our food, how we store our food and our water. We can also watch out for donors during blood donor screening. Also, we can isolate those newborns and children that could have acquired the disease from their infected mothers. Also, wearing protective clothing which is neutral colored and medium weight, because as an interesting fact is that the SCC flies, they can bite through thin layered clothing and they are attracted to brightly colored material. We can also avoid bushes or areas where there's a chance of bugs and flies being present. And also a very helpful one is that we can use insect repellent. And that's all we have for this presentation. You will find the reference link in the bio of this video. And if you happen to have any further questions, you can email me at the following email. Do subscribe to our YouTube channel. We are trying to help you guys as much as you can, as well as learn along in the process. Thank you all for listening and have a great day.